What is a control group? A control group is a set of subjects in a scientific experiment who do not receive the treatment or intervention being studied. In experimental design, researchers typically divide subjects into two groups, a control group and an experimental group. The experimental group receives the treatment or intervention being studied, while the control group does not. Control groups serve as a benchmark for comparison against the treatment group, allowing researchers to determine if the intervention has a measurable effect. Control groups are fundamental to the scientific method and experimental design, helping to isolate the effects of the variable being tested. Scientific conclusions are drawn by comparing the outcomes between control and experimental groups. Why do control groups matter in scientific research? Control groups establish a baseline for comparison, which is essential in research. They serve as a reference point showing what would happen without any intervention. Let's compare experimental and control groups. The control group receives no intervention, while the experimental group receives the treatment being studied. Determining causality is impossible without control groups. Researchers need to distinguish whether observed effects are due to their intervention or would have occurred anyway. A before and after comparison helps demonstrate this concept. In the control group, measurements before and after show minimal natural changes, while the experimental group shows notable differences that can be attributed to the intervention. Without control groups, scientific research fails in several critical ways. Researchers cannot establish causality, rule out placebo effects, account for natural progression of conditions, or detect unrelated environmental factors that might influence results. Confounding variables are factors that might influence the experimental results besides the treatment being studied. When we conduct experiments, we want to measure how a treatment affects an outcome. However, confounding variables can interfere with our results by influencing both the treatment and the outcome. Control groups help isolate the effect of the independent variable by ensuring all other conditions remain constant. Let's consider an example where researchers study the effect of a fertilizer on plant growth. Without a control group, they might observe that plants with the fertilizer grew 30% more. However, what if there was a hidden confounding variable, such as uneven sunlight exposure? With a control group that is identical in all aspects except for the fertilizer, we can isolate the treatment effect. Both groups receive the same amount of sunlight, soil, and water, but only the experimental group receives the fertilizer. Now we can accurately measure the effect. If the experimental group grows 30% and the control group grows 20%, then the actual effect of the fertilizer is only 10%. Bias in research can significantly impact the validity of scientific studies. There are several common types of bias in research. These include selection bias, confirmation bias, and observer bias. Selection bias occurs when participants selected for a study don't truly represent the target population. This can happen when certain groups are overrepresented in the sample. Control groups help reduce selection bias through random assignment. By randomly assigning participants to either the experimental or control group, researchers ensure both groups represent the overall population. Confirmation bias is the tendency to interpret data in ways that confirm existing beliefs or hypotheses. Researchers may unintentionally focus on results that support their hypothesis while dismissing contradictory evidence. Control groups help reduce confirmation bias through blinded studies. When researchers don't know which group received the intervention, they can't unconsciously favor data that supports their hypothesis. Observer bias occurs when the researcher's presence or expectations unconsciously influence participant behavior or how data is recorded. Participants may try to please researchers by giving expected responses. Control groups help reduce observer bias through double-blind studies. when neither participants nor researchers know who's in which group, the influence of expectations is minimized. Effective control groups are crucial for scientific validity. They must have several key features to ensure proper comparison with experimental groups. First, control groups should receive either no intervention or a standardized treatment like a placebo. This establishes the baseline against which experimental effects are measured. Second, control groups must experience identical environmental and experimental conditions as the treatment groups. This includes factors like temperature, timing, handling procedures, and measurement methods. Third, control groups must come from identical populations as the experimental groups. This is especially important in biological research, where genetic variation can significantly impact results. Finally, the control and experimental groups should differ only in the specific variable being studied. This isolation ensures that any observed effects can be attributed directly to the test variable. When these four key features are properly implemented, control groups establish accurate baselines, isolate experimental effects, ensure scientific validity, and enable proper statistical analysis. Remember, effective control groups receive no intervention or a standardized treatment, experience identical conditions as treatment groups, come from identical populations, and differ only in the variable being studied. Let's examine how control groups are used in enzyme activity assessment. The starch iodine test is commonly used to assess enzyme activity, particularly for enzymes that break down starch. Our experiment uses a starch solution, iodine reagent, which turns blue-black in the presence of starch, and the enzyme amylase, which is only added to our experimental group. At the start of our experiment, both the control and experimental beakers contain starch and iodine, which creates a blue-black color. After 30 minutes, we observe the results. The enzyme in the experimental beaker has broken down the starch. Let's interpret these results. In our control beaker, which contains no enzyme, the blue-black color remains, indicating starch is still present. In the experimental beaker, the solution has turned yellow, showing the enzyme has broken down the starch. The control group is crucial in this experiment. It confirms that the color change is due to enzyme activity, not spontaneous starch breakdown. It also allows us to quantify the enzyme's effectiveness by comparison. Scientists can precisely measure enzyme activity using colorimetric analysis, spectrophotometer readings, or by tracking the rate of color change over time. This enzyme activity assessment demonstrates the essential role of control groups in biochemical experiments. Controls help isolate the variable of interest, in this case enzyme activity, and provide the basis for quantitative measurements in enzyme kinetics. Without the control group, we couldn't confidently attribute the color change to enzyme activity. In pharmaceutical research, control groups are essential for determining if a drug truly works. A typical drug trial divides participants into two groups: a treatment group that receives the investigational drug and a control group. The treatment group receives the actual medication being tested. The control group receives a placebo, typically a sugar pill that looks identical to the real treatment but contains no active ingredients. The placebo effect is a fascinating phenomenon where patients can experience improvements simply because they believe they're receiving treatment. This graph shows how both active drugs and placebos can lead to improvement over time. The difference between the two lines represents the placebo effect, improvements that happen just by believing in the treatment. This is why it's crucial to account for the placebo effect when testing new drugs. Without a control group, we might attribute all improvement to the drug itself. Let's look at a simplified clinical trial design to see how control groups are implemented. After patients are recruited, they're randomly assigned to either the treatment group or the control group. In a double-blind study, neither the patients nor the researchers know who is receiving the real drug versus the placebo. Data is collected from both groups, measuring the same outcomes to determine if the drug outperforms the placebo. Finally, statistical analysis compares the results between groups to determine if any observed benefits are statistically significant and not due to chance. To summarize, control groups in drug trials are crucial for several important reasons. They help isolate true drug effects from placebo effects. Control groups account for natural healing processes and statistical regression to the mean. They help researchers distinguish between actual side effects and coincidental symptoms. Most importantly, control groups provide scientific validity to the drug approval process. Statistical analysis is where control groups truly demonstrate their value in research. Control groups provide a crucial reference point for statistical comparison. Statistical significance helps researchers determine if observed differences between groups are meaningful or simply due to random chance. When we collect data from both control and treatment groups, we typically see distributions like these. The p-value represents the probability of observing the data, or more extreme results if there were no true difference between groups. Researchers typically use a threshold of 0.05. When the p-value falls below this threshold, the results are considered statistically significant. Let's look at some practical examples of how statistical analysis is used with control groups in different research contexts. In a drug trial, a p-value of 0.02 indicates strong evidence that the treatment has a real effect compared to the control group. For an educational method study, a p-value of 0.72 suggests no meaningful difference between the treatment and control groups. In agricultural research, a p-value of 0.4 provides evidence that the treatment increases crop yield compared to controls. Proper statistical analysis of control group data provides several benefits for research. It validates experimental findings, helps determine practical significance, and guides future research directions. In the final section of our discussion on control groups, we examine the important ethical considerations researchers must address. Ethical challenges are particularly pronounced in medical research. When control groups are used, patients may not receive potentially beneficial treatments. This raises concerns about withholding the standard of care, especially when working with vulnerable populations. The central ethical question is it ethical to withhold potentially beneficial treatments from the control group? This creates tension between scientific rigor and patient welfare. To address these ethical concerns, researchers have developed alternative study designs that maintain scientific validity while reducing ethical dilemmas. Crossover studies allow each participant to receive both the treatment and control conditions at different times, separated by a washout period. This ensures everyone eventually receives the active treatment. Adaptive trial designs allow researchers to modify treatment allocation based on interim results. This approach increases the likelihood that participants receive effective treatments while reducing exposure to inferior options. Despite these challenges, well-designed control groups ultimately protect future patients from ineffective or harmful treatments. Researchers must balance scientific rigor with ethical considerations. Independent ethics committees should review all research protocols, and informed consent is essential. The ultimate goal is to maximize benefits while minimizing harm to participants. While ethical considerations must be carefully addressed, properly designed control groups ultimately help ensure that treatments are truly safe and effective for all patients.